Hello everyone, welcome to this webinar as part of our 2021 Analytical Extravaganza. I'm Faith Hayes, LCMS Sales Specialist at Shimadzu Scientific Instruments. I will be your moderator today, and we will be hearing from Volker Bornemann and Darren Stevens. Our topic is Applications of ICP-MS in Bioanalysis and the Life Sciences. Before we start, I want to share a couple of notes for our viewers. The webinar console has a variety of items to help enhance your experience and interaction with us. In the screen, you will see the following items. The slides will appear on the left-hand side. Directly under the slides, you will see a resource list with clickable links relevant to the material being presented today. On the top middle is the widget for questions and answers. Please submit your questions during the presentation through this widget, and we will answer them during the Q&A session. Just below the Q&A box are the moderator and speaker bios. You may expand the items here to learn more about us. On the right are survey questions that you may fill out anytime during or after the presentation. Finally, at the bottom pane are the icons to bring up all of these widgets in case they are minimized or hidden. All right, without further ado, let's get started. Again, if you are just joining us, I'm Faith, your moderator. Today we will be hearing from Volker Bornemann and Darren Stevens, and our topic is Applications of ICP-MS in Bioanalysis and the Life Sciences. A brief bio about Volker. In 2014, he founded Avazyme Incorporated, a contract research organization that provides customized field and laboratory testing services for a variety of clients from agriculture and the entire food chain, ag tech, pharmaceuticals, cosmetics, and more. Volker is also a member of the North Carolina Biotechnology Center Ag Biotech Advisory Council. Prior to starting his own companies, he was the president of Smithers Viciant, a mid-sized contract research organization focused on environmental testing. He began his career at BASF in Germany, where he held various R&D leadership and operating roles, some of them in the U.S., in the Agricultural Solutions Division. He was involved in innovation teams and regulatory science with leadership roles in the OECD and international regulatory task forces and other industrial associations such as ECPA, CLA, CLI in Europe and the US. When not in the office or traveling on business, Volker is teaching and mentoring students as an adjunct professor at NC State University. He can also be found roaming the great outdoors, either skiing or on foot with his dogs or in his kayak. Volker received his master's and PhD in chemistry from Rice University in Houston, Texas, studying the biosynthesis of antibiotics from fermentation. In addition, he received a postdoctoral fellowship at the University of Hawaii at Manoa, where he studied the biosynthesis and novel structural elucidation of cyanobacteria. Darren Stevens is a research scientist at Avazyme. Darren has over 20 years' experience working in biochemistry, bioanalytical, and contract research laboratories. He has extensive experience in mass spectrometry and method development. Darren received a master's degree from the University of Texas at Austin. Now I will hand it over to Dr. Volker Bornemann.
Well, thank you, Faith, for the kind introduction. We also have a couple of introductory slides, which I'll go over quickly. My name is Volker Bornemann. I'm a chemist by training. Got my PhD from Rice University in Houston, Texas, and then worked for 22 years at BASF in the Agricultural Solutions Division, mostly doing analytical chemistry and regulatory science. I later on joined Smithers Viciant as their president, and there I learned a lot about environmental science and also how to run a business, which then I decided to do on my own. I started Avazyme as a company in 2014 here in Durham, North Carolina, in Research Triangle Park. And in my spare time, I'm also an adjunct professor at NC State University in the chemistry department.
I'm Darren Stevens. I am also a chemist and biochemist by training. I have a BA in biology and an MA in molecular biology from the University of Texas at Austin. I've worked in university labs doing research and as well have been in the industry for over 10 years. I've worked at Enthalpy Analytical, Tandem Labs, Covance Labs prior to working here. In a variety of roles as a LC-MS analyst and method development chemist, as a project manager and principal investigator, and as well as doing a lot of instrumentation and IT maintenance and support. So currently I'm a research scientist here at Avazyme and also an instructor at Durham Tech Community College part-time.
Here at Avazyme, we're having a lot of fun providing customized solutions for our customers from agriculture and the entire food value chain. So we start with input parameters like doing customized research on pesticides and with ag biotech companies nationwide and even from abroad. We were working directly with farmers, growers. We work with food processors, beverage processors, brewers, distillers, and packaging companies. We work for distributors, importers, exporters, and sometimes also for retailers and consumers. A lot of applications for ICP-MS spec are actually found in the pharmaceutical value chain. So we can investigate medicinal herbs, for example, for their heavy metals content, but also for their micronutrient content. We have one customer who makes vaccines out of tobacco, and they make actually a whole variety of vaccines, like Ebola vaccine. They have flu vaccines, and most recently, of course, they also are working on a COVID-19 vaccine. It's very important that the heavy metals are monitored in the crop that is used to make these vaccines, because you don't want heavy metals to get into your body through the vaccine product. So this particular client makes very sure that there are no detectable levels of heavy metals in their crop. The same is true for customers of ours who make biological pharmaceutical drugs by a fermentation process. And here too, they have to be concerned that some of the ingredients that are being used to facilitate the fermentation process are not contaminated with heavy metals. And this includes, for example, the water that's being used, and sometimes comes from city water, and their heavy metal content is important to monitor, but also potential intake of chlorine in unacceptable levels, because a lot of the city water is chlorinated. But there also are other ingredients that are being used in the fermentation processes. For example, corn steep liquor as a carbohydrate source, and that can come from field-grown corn, which in turn may have picked up heavy metals from the soil or from the irrigation water. So it's important to monitor for heavy metals in all the excipients, in all the intermediates, and in the final products, to make sure that there are no undesirable contaminants in these products. We also provide a lot of services for the hemp value chain. which very nicely demonstrates the different types of investigations we can do. So we support the hemp farmers by analyzing their soils and irrigation water, in particular for heavy metals, that we can do by ICP-MS spec, and also micronutrients. And we support them with their entire process chain to either crude extract, smokable flour, or isolates of certain cannabinoids. We perform our testing according to certain quality standards, including ISO 17025, and this includes our ICP-MS spec type of investigations. We are following guidelines from the USDA. We are USDA APHIS certified and permitted. We have a DAA permit for controlled substances, and we have operating procedures in place for all the different types of tests that we use, including for ICP-MS spec, which we utilize in GLP studies, but also for GMP analysis to support pharmaceutical production facilities. So let's talk about ICP-MS spec. How does it actually work? What you see depicted here on these pictures is actually our instrument in our lab, equipped with an autosampler. And I'm also showing you the Mark 6 microwave digester on the right-hand side that we will talk about in more detail.
In the process ICP-MS, the samples are infused into the instrument and pass through an inductively coupled plasma that's generated from argon gas. And from there, the elements are broken down into atomic and small polyatomic ions. And from that point, they pass through the cones into the mass spec and are read on a detector. Proper breakdown of the sample is crucial to successful ICP-MS, so we employ microwave digestion. It uses reusable vessels, and we add sample, acids, and a mix of that and water to add to the vials, cap them, put them in the microwave, and they run for a specific amount of time at a specific temperature, and that allows the sample to be physically broken down. Sometimes some pre-digestion in the acid prior to addition of water is necessary to ensure full digestion, but you should receive a clear or translucent digest at the end, and then you volume that up with water to allow dilution of the acid so that it becomes more compatible with the instrument, with the ICP-MS. The acid digestion is done in inert metal-free tubes, and we can do 40 tubes in this instrument with our setup at a time. And this is one of the main methods to be used because it's fast, it's efficient, and it's quick.
Yeah, it's also the industry standard. You find this particular digester described in several of the EPA methods for heavy metal determination and other metals in the environment and water. And CEM actually has an amazing selection of application notes. It's a, believe it or not, a 582-page document that gives you a lot of different scenarios for sample prep, different types of samples in this digester.
This slide shows a periodic table with the elements that can be measured by ICP-MS highlighted. and most of these can be measured with accurately below 10 ppb, and almost all of them can be measured in the same run. So you can measure 20, 30, 40 of these all at once in the same run. Since you're infusing the sample, the mass spec just takes a little bit longer time to measure when you're depending upon the number of analytes you're trying to get. So a 4 analyte heavy metals run will take a far shorter time than a nutrient run, which may be measuring 30 different analytes. But the beauty is that you can get all of this information in a single run of sample. We also typically measure multiple replicates of samples and report the average. Typically we report 3 replicates here, but that's customizable as well in the software. For our typical heavy metals analysis, we measure arsenic, cadmium, mercury, and lead. We digest the samples and quantify those against calibration standards that are prepared with certified components. For cadmium, mercury, and lead, we measure multiple isotopes. They give us extra data for interferences and other kind of quality control measures, as well as just allowing us to check the system. The isotopes are monitored for confirmation. And then we have guidelines on which of them to report, and in case of lead, we'll report them summed. We use internal standards. In our case, we typically use thallium and yttrium, and we infuse those in tandem with the samples. So these methods are used in, this is used for compendial methods such as USP39 or Cresta number 93. So the heavy metals that we can measure are typically done to meet a regulatory requirement or monitoring requirement. FDA has several requirements, say, for arsenic in apple juice, but it is not heavily regulated. This is not a heavily regulated area for FDA, and also even for the states, the monitoring levels and acceptable levels of heavy metals vary from state to state. For instance, with hemp, that's just now coming together, and so the need to test these and test them for varying regulatory environments is important. So to make sure that we give the best data possible, we try to follow the FDA guidance on reporting and monitoring the different isotopes as listed here on this slide. So for most of these, there's a single isotope monitored, but for some, there's multiple isotopes which are monitored, and one of them is reported. In the case of lead, though, we monitor 3 separate isotopes, 206, 207, and 208, and report the sum. There is a 4th isotope of lead, 204, which is typically not monitored or reported due to its low abundance. But this is an ever-changing environment, and the ability to measure these accurately is very important for our customers meeting their regulatory requirements. Okay, and this slide shows a screenshot from the Lab Solutions ICP-MS software. So this is a layout of the acquisition mode with instrument controls shown on the right-hand side, instrument status on the right-hand side, the center top frame is the spectrum being currently acquired. The center left frame is where you enter sample information. That will be volumes and weights and all of those things, which are used to compile the data, which is actively calculated in the lower center frame. To the right of that is the individual data. In our case, we replicate 3 runs of the data, and so all 3 individual replicates are shown to the right, and the average is shown in the left frame there. So it's really nice that it's calculating the data as you do it. There's no waiting for the end of the run to actually get an idea of how things are running. So the software is very easy to use. We've had really no issues with it whatsoever. It's been really easy to set up, really easy to use. I definitely recommend that. So the Lab Solution software being so easy to use is actually fairly quick in order to get other people up to speed on. So after the other thing you can look at after the analysis is the calibration. So you have on the lower part of the panel, you can see that there are tabs, and on the calibration tab, you can see the plots of all of the calibration standards. So this is one of our recent runs of the heavy metal analysis, the arsenic, cadmium, mercury, and lead analysis, and you can see all of the curves on this screen. In this case, we've got arsenic selected, so it's showing larger on the right-hand side, and you can see all the individual replicate data down below that. And then on each of the others, you can see summaries in the rest of the window. And this slide shows the Lab Solutions ICP-MS software in the profile viewing mode, where you can see the spectrum and zoom in on the different ions and even see the map of the natural abundances, and use that as an identifier, as well as use it to see possible sources of contamination or possible sources of interference. The software actually has a lot of very good built-in correction factors. So, and a lot of them are automatically applied, but you have a large degree of latitude on which of them that you apply on any given case. In this case, I'm looking at lead, and with the 208 ion highlighted. And so in the top frame, you see the entire spectrum. The yellow and blue areas are the areas of interest that we're in, and the center frame shows the individual ions, and I have what's called an isotopic ratio selected there, and so it's giving me a color-coded illustration on the peaks to show me what the natural abundance should be for that, would look like for that ion, so that I can use that to help positively identify it, but also give me an idea if there's another interference or something, and which I can investigate on the lower frame, and I can choose several common variants to show there to investigate. So the tools in this section of it are very robust and very user-friendly. And so on the right panel there, you see all of the available ions and isotopes for this particular element, and the natural abundances and what to expect. So this profile mode is very, very good for positive identification. In ICP-MS measurements, the sample integrity is crucial, and the environment is almost your enemy here because metals are everywhere. So contamination is not difficult to pick up. So it's important to be very careful with sample handling so that we don't measure anything with metal utensils. We avoid glass. The glassware that a sample may be contained in or transferred to can contain various metals depending upon how it was produced, where it was produced, what other processes it goes through. So we try to avoid glassware. Plastic generally has a lot lower metal contamination. And any of the reagents that touch the sample are also crucial. So any of the acids that we use for digestion are trace metal grade that are especially low in any of the metals that we measure. We also use high purity reverse osmosis water, but for the same reason that we prove that it is low in metals contamination or absent, and that we check it constantly. Dust is another big source of potential contamination. So we do a lot of things in the fume hood where the air is moving, and that helps to keep that down. And as we work with the samples, we're very careful not to introduce any outside sources of metals. So we're careful with transferring, and that other extraneous sources are minimized as best we can, because metals stick around forever. They do not degrade, and so storage stability is not a problem when it comes to a storage study. What's more important is accumulation. So if it's leaching from the storage container that you're performing a storage study in, then you can increase toxicology because of the cumulative effect and the fact that these metals are everlasting. And so we always are on the lookout for contamination, and it can come from anywhere. That is.
Metals analysis is particularly important to ensure product safety of all sorts of different products, including food products, but also other consumer products. So where do we find these heavy metals? So arsenic, for example, is found a lot of times in rice, where it also can bioaccumulate. So there, a number of years back, rice has been in the press, and it's also on the radar screen of the FDA to make sure that the rice does not contain unacceptable level of arsenic. Apple juice is another one where sometimes arsenic is found. How does it get in there? Sometimes through filtering media like diatomaceous earth can contain high levels of arsenic depending on where the diatomaceous earth has been mined. Cadmium is of concern. A lot of cadmium finds its way into landfills from where it leaches into the groundwater. The main source is the rechargeable nickel-cadmium batteries that once disposed of improperly in landfill can lead to contamination in the environment. Same with mercury. Mercury can be found in high levels in water, but it can also find its way into other consumer products. It used to be in use in mercury switches in metal alloys, in thermometers, in other types of meters like your blood pressure meter. In doctors' offices used to have mercury-filled scales. And of course, lead is found abundantly in nature. A lot of it, unfortunately, comes from hunting activities. In particular, ponds have been contaminated with lead shot from duck hunting activities, and we find it in the water, but also in the water sediment system. Chromium can be of concern. Aluminum is also oftentimes brought into context with Alzheimer's disease. Barium, I'm not sure how barium winds up in nature. The only use of barium that used to be quite common is as a contrast medium for X-ray diagnostics. But there it is applied to the patient in a completely water-insoluble form as barium sulfate, and is passed through without bioaccumulating in the body. And last but not least, nickel. Some people are very allergic to nickel, and nickel is found in a variety of alloys, and that's how the American coin got its name. Yeah, another big area of concern with respect to heavy metal contamination and consumer safety is the heavy metal content in vape products and smokable products. So you can find all of the metals listed on the left side in these products, and we can measure them to trace levels with the help of ICP-MS and in conjunction with the appropriate digestion method in our microwave digester. We find significant amounts of heavy metals in tobacco-based products, where the tobacco has pulled the heavy metals from the soil, or sometimes it comes in through the irrigation water or runoff from neighboring fields, and or if the irrigation is happening with pond water that may be contaminated with lead, high levels of lead from prior duck hunting activities. We also find it frequently in hemp-derived products because hemp is an excellent bioremediation crop and will pull out all sorts of stuff from the soil and also pick up stuff from the irrigation water. So the same principles apply. We can measure them down to very low levels in the hemp products, whether it's tinctures or even isolates. Sometimes we find the heavy metals, but also in particular in the smokable flour. And why is the smokable products, why is the heavy metals content so important? Well, if you think about it, the lung is designed to ingest or incorporate products from the air very efficiently, and it's designed to do that mostly for oxygen, but through the same route you can get other products very quickly into the bloodstream, into the body, and in the case of heavy metals, they will bioaccumulate there. So they will, once they get into your body, they will stay around for a very long time. And you can use ICP-MS, for example, to determine heavy metals in the body by investigating hair. We can also examine human tissue samples for heavy metals content, and this is frequently done in forensics when there is a suspicion that a heavy metal, in particular arsenic, may be responsible or lead poisoning may be responsible for certain observed toxicological effects. Many products have not been studied extensively through inhalation toxicology studies. These studies are very complicated, complex. There are only very few toxicology labs that can perform these studies. And a lot of the testing is done more on the exposure route and exposure potential, in particular for vape products and smokable products. Elemental analysis by ICP-MS is also important in order to assess product quality. So in one way this can be done is through process troubleshooting and optimization to minimize the amounts of heavy metals that find their way into food or pharmaceutical products. So excipients, for example, that are being used to make pharmaceutical products are always investigated for potential heavy metal content. So unacceptable heavy metal amounts have to be avoided, and ICP-MS can ensure that heavy metals are not contained in the products or in the process to any significant extent. It's also important in the context of plant health. So with the help of ICP-MS spec, we can determine the levels of micronutrients in plants, in the soil. We can assess how minerals are being taken up by plants. Sometimes this can be facilitated through certain soil microbes and the microbiome of the plant. And there are ag-tech companies that actually are making products that help plant to be healthier, stronger, and more nutritious by maintaining the appropriate micronutrient levels in the crops and in the soil. Minerals are also very important for human health to make sure we don't get excessive amounts of certain undesirable metals into our bodies, but maintain appropriate levels of certain desirable minerals through our food and beverage intake and water intake. So here iodine, of course, is important, but also iron, selenium, zinc can help to improve human health. The same is true, by the way, for animal health. Here, some of these metals and minerals play an important role in animal health as well. We also see the advancement of certain organic pesticides. These are usually copper-based or sulfur-based. And here, for some of these copper-based pesticides, ICP-MS can actually be used to assess the concentration of what we call the active ingredient. In this case, it's either copper, it's usually copper salts with various counter ions for the copper to make it longer lasting usually as a fungicide in crop protection. And there are a lot of these copper-based products and sulfur-based products are listed as acceptable pesticides for organic production. But we also find them to some extent in all-natural diatomaceous earth, which is oftentimes used as a filtering agent in fruit juice production to clarify your apple juice or other fruit-derived juices, as well as they are used in wine production, in beer production. And through this diatomaceous earth, depending on where it comes from, where it has been mined, it can have high levels of arsenic and even mercury, and through that contaminate the fruit juices or other beverages. So ICP-MS can measure the levels of these undesirable heavy metals in just about any food and beverage matrix that you can think of. And thus, ICP-MS spec makes a very important contribution to food safety, but also food quality. So that brings us to the end of our formal presentation. I hope Darren and I were able to give you some good examples of applications of ICP-MS in the various scenarios that we described and the various areas where ICP-MS can be a very valuable tool to provide information about metal content of products, of crops, of soil, water, and other areas of concern. So ICP-MS has been one of the main workhorses in our laboratory. I can say we are extremely happy with the Shimadzu equipment. It is very easy to learn how to use. It's very easy to maintain. It's very easy to clean out. Our instrument is pretty much running 24/7 and is one of our main workhorses in our laboratory. So I hope we gave you an interesting overview of what ICP-MS can do, and we are happy to answer any questions you may have at this point on how ICP-MS may be helpful for you.
Let's thank Volker and Darren for such an exciting presentation. Again, under the resource list, you can find more information about our analytical solutions. If you have not had a chance, please fill out the survey questions on the right-hand side to provide feedback and request additional information. We also encourage you and your colleagues to attend our upcoming webinars, which encompass a wide variety of application areas. The registration link is located at the bottom of this slide. At this time, we will begin our Q&A session. Thank you to the audience for attending and sending questions. We have time for a few questions. First question, Volker. For the hemp data, was that in the flower, the analysis that you guys did? And if so, have you looked at other parts of the plant like the leaves or the stem?
I believe, but Darren can confirm that. I think that particular analysis was done on hemp flower, where usually the concern is the greatest, especially if it's smokable flower. But we have looked at other plant parts in the past, and as you know, hemp is a very good bioremediation crop. So you can actually use hemp to clean up contaminated soils, because it will pull anything that it can get its roots on out of the soil and store it in the various compartments in the plant. So, for example, we find heavy metals in the leaves, actually more so than in the flower. The leaves are probably the largest storage space for heavy metals, but we also find it in stems and roots.
Great, thank you. What is the role of the ICP in ICP-MS?
Well, it's our main detection tool when we are looking for the presence or absence of metals and heavy metals. And ICP is really the best and most sensitive way to measure those that I'm aware of.
Great, thank you. When analyzing the vaping samples, are you guys analyzing the actual vapor sample, or are you analyzing the liquid?
We are, but sometimes there are questions about storage stability, so the vape cartridges have metal parts on it that may get in touch with the actual vape liquid. And then we look over we look at what extent the metals may find their way from the cartridge itself into the vape liquid.
So then you're analyzing what part? I'm sorry if you said it and I'm just missing it.
Yeah, no, we are analyzing mostly the liquid, the vape liquid.
The vape liquid, okay. Great. Sorry, just getting back to the list. How does the mini torch reduce the amount of argon required?
I think it's just a smaller diameter flame, so the plasma is smaller than you will find in older instruments from other instrument manufacturers. Miniaturizing the argon stream and getting away with essentially less argon flow significantly reduces the argon amounts that you need to run the instrument. So it makes it, argon is not a cheap gas to buy. It makes it, especially at the purity grades that we needed for ICP mass spec. So it minimizes our operating costs.
Okay, great. You mentioned about arsenic levels in rice. Do you measure the inorganic specimen of arsenic by LC ICPMS?
We do not. We don't have an LC hooked up to the ICP because usually the question is not in which type of species is arsenic bound. So through our digestion process, we pretty much chop all the organic molecules apart and free up the arsenic in its free form, which we then can quickly detect as full arsenic, if you want, by ICP mass spec. If you wanted to know which exact arsenic organic species is present in the original crop or soil, then you would have to separate them out by liquid chromatography first.
And then analyze the fractions, essentially, from the LC.
Right. You would probably do it online. So I've seen in other labs, they had an LC in front of the ICP mass spec. And but for the questions that we need to answer for our clients, it's usually not the demand.
Got it. Thank you. How does the CEM handle silica in matrices such as plant material? The follow-up comment being that that nitric acid doesn't generally break down silica.
Yeah.
Yes, we mentioned the CEM digestion guide in one of our slides there, and that's available on their website. And that's a really great resource in adding additional acids or different tips to digest a wide variety of different matrices. So for most of the plant material, nitric is pretty sufficient, depending upon the way that you tweak the procedure. But yes, for other things like soil, we have to add in things like sulfuric acid or hydrofluoric. But that guide has been invaluable to us. It's just a very, very large number of recipes.
Great. Thank you, Darren. Next question. What dictates the detection or quantification limit in ICP-MS? And are there specific techniques that can be used to optimize the quantification limit?
Probably the detection, the biggest factor is going to be your background level of any given metal. So you can moderate this by using the cleanest reagents that you can, the cleanest labware that you can, to bring it down as low as you can. So the lower you get your background, the lower level you'll be able to see above that background.
Yeah, and on the chart that we showed, which actually was created by Shimatsu, it gives different sensitivity levels for the different metals that you can analyze, or the different elements, I should say, that you can analyze. So different elements have different, I guess it's ionization potential, and that determines the sensitivity, of course, together with the sensitivity of your mass spectrometer. And what also can work against you is if a particular metal has a number of isotopes, as we showed you for the example of lead, where you have theoretically 4 measurable isotopes, but one of them only occurs at 1%, so you don't detect that to very low limits. And so in a sense, this isotope, the presence of different isotopes with different natural abundance also drives down your sensitivity.
Great, thank you. Are there calibration curves for any elements nonlinear?
The only one that we really sometimes have to change it to nonlinear is mercury. But, you know, mercury has its own issues as far as carryover and everything. So if we go to really high levels of mercury on a curve, it will tend to lose linearity. But to battle the carryover, we've actually cut back on the mercury in our curves so that mercury does not go to the full range that the others do. So that makes our curves more linear.
Great. And as a follow-up, how do you deal with the mercury carryover? Are there any specific tips or tricks that you guys use?
The first was to cut back on the mercury in our curves based on the samples that we were receiving. We started out with a curve from 1 to 10 ppb for the plant material, and we since expanded that range up to 50 ppb for arsenic, cadmium, and lead. But at mercury, I've actually cut it back to 5 to help cut back on the carryover, because we're not seeing samples routinely that are above that level.
Great,
so
thank you.
The main thing is lots of blanks and trying to only use as much mercury as you have to for your curves.
Okay, good. Thanks. trying to get my pronoun right. Has Avazyme run any tests under USP 232?
Well, yes, we have for our pharmaceutical clients, essentially. They typically ask us to run either customized tests that they customize specifically for their particular drugs and drug formulations, or they refer to the USP methods to measure the metals.
Thank you. And then the last, we just have a few minutes left, but there's a few questions that came in just about the instrument maintenance. One of them is, do you need to break vacuum to clean the skimmer cone or sampling cone?
No, you do not. So that makes daily cleanup very easy.
Good. And do you have to perform daily cleanup when you're running in a high throughput fashion?
It depends on the. We kind of look at what type of samples we're running. Plant matrix samples tend to be a little dirtier, so if we're running, if we've had a full day, we'll definitely need to clean the cone. If they're kind of, let's say if it's a lot of water samples, things that don't have a lot of organic matter in there, we might be able to get away with skipping a day. But we try to make sure that the cone is in pretty good shape before any run starts. So, daily is what's recommended by Shimadzu, and we try to hold to that as much as possible.
How long does it take you to do the cleanup?
The cleanup is somewhere around 20 minutes. That includes a 5-minute soak in acid, and then it's just the rinsing and reassembly. So it's something that's easily built into the setting up the instrument during the day.
And do you need any special tools to perform that maintenance?
Those are all supplied with the instrument. It's essentially a hex wrench. A single hex wrench is all you really need. There are 4 screws that hold each of the cones on, and that's all you have to do to remove it. Gasket behind the large skimmer, behind the big cone there, and other than that, it goes pretty quickly. The acid does most of the work. The acid soaks, so there isn't a lot of manual scrubbing or anything to do.
Great, thank you, Darren.
And also, there's a pretty detailed procedure for how to do that. That's included with the instrument. Lots of really nice pictures and everything. So it's
great, thank you. It looks like we are at the end of our allocated time for the webinar, so I would like to thank the audience for all of the interesting questions. If there are any questions that were unanswered, we will reach out individually to each of you for those questions that we were not able to answer today. Once again, thank you for attending and participating. We will send you an email with a link to view a recorded version of this presentation anytime. Again, we encourage you and your colleagues to attend our upcoming webinars, and the registration link is located at the bottom of this slide. Have a great day. Bye-bye.